Hello everyone. In the earlier class, we had discussed about kidneys, the primary organs of the urinary system. There, we had dealt with its gross anatomy and internal structure. So now, we'll look into the nephrons in detail. A nephron is the basic structural and functional unit of kidney. There are about 1.2 million of nephrons in each kidney in human beings, which start to decline after the age of 45 years. The nephrons are hollow tubes through which the blood is filtered and modified to finally form the urine. The total length of the nephron ranges from 45 to 65 millimeter. Now, the parts of nephron. A nephron consists of the blind end portion called renal corpuscle and the tubular portion called renal tubule. A renal tubule is formed by proximal convoluted tubule, a loop of Henle which has a descending portion and an ascending portion, then distal convoluted tubule and the collecting duct system. Each of this segment is formed of single layer of epithelial cells placed on the basement membrane, which are modified accordingly to perform their specific functions. Renal corpuscle or Malpighian corpuscle is a spheroidal and slightly flattened structure with a diameter of about 200 microns. Its function is to filter the blood which gets eventually transformed into urine. Glomerulus and Bowman's capsule together form the renal corpuscle. Classification of nephrons. Based on the position of renal corpuscles, nephrons are classified into two: cortical nephrons and juxtamedullary nephrons. These cortical nephrons or superficial nephrons are those whose renal corpuscles are situated in the superficial layer of cortex, that is the outer cortex, whereas juxtamedullary nephrons are those whose renal corpuscles are situated nearer the corticomedullary junction or the inner cortex. There are many differences between the cortical and juxtamedullary nephrons. Let us consider them one by one. First one is the location of renal corpuscles that we have just discussed. That is, the cortical nephrons have their renal corpuscle in the superficial cortex, and juxtamedullary nephrons have their renal corpuscles in the inner cortex or nearer to the corticomedullary junction. About 85% of the total nephrons is formed of the cortical nephrons and 15% accounts to the juxtamedullary nephrons. Length of loop of Henle is shorter for cortical nephrons and longer for juxtamedullary nephrons. I had told earlier that the loop of Henle is having a descending limb and an ascending limb. Irrespective of the type of nephron, the descending limb will be thin in all the nephrons. That is, both cortical and juxtamedullary nephrons will be having a thin descending limb. But there is a slight difference in case of ascending limb. That is, the cortical nephrons have a thick ascending limb, but the ascending limb of the juxtamedullary nephrons have both thin segment and thick segment. So, the loop of Henle of the cortical nephrons will be having a thin descending limb and a thick ascending limb, whereas the juxtamedullary nephrons will be having a thin descending limb, a thin ascending limb and a thick ascending limb. Okay. And since this thin ascending limb is absent in cortical nephrons, the length of the thin limb will be shorter and it will be longer in juxtamedullary nephrons. Now, the size of the glomerulus. It will be small in cortical nephrons and large in juxtamedullary nephrons. The rate of filtration is higher in juxtamedullary nephrons. Efficiency in concentrating urine is more in juxtamedullary nephrons. The renin content is more in cortical nephrons, and the main function of the cortical nephron is urine formation, and the extramedullary nephron is urine concentration. Another point is regarding the efferent arterioles. The efferent arterioles of the cortical nephrons form peritubular capillaries that surround the segments of the nephron of its own and of adjacent ones, whereas efferent arterioles of the extramedullary nephrons form peritubular capillaries and an extra set of capillaries called vasa recta. The efferent arterioles in cortical nephrons form peritubular capillaries, whereas the efferent arterioles in juxtamedullary nephrons form peritubular capillaries and an additional capillaries called vasa recta. These vasa recta are straight, long capillaries that run deep into the medulla, having descending and ascending limbs. And we'll learn about this in detail in the upcoming sessions. We will look into the structure of nephrons in detail. First is the ultrastructure of renal corpuscle. We all know that renal corpuscle consists of Bowman's capsule that contains glomerulus and mesangium. So here the first step in urine formation takes place. There are three major steps in urine formation. They are ultrafiltration, tubular reabsorption and tubular secretion, of which the ultrafiltration, the first step in urine formation, occurs in the renal corpuscle. Okay, so what is a glomerulus? Glomerulus is a tuft of capillaries located in the initial dilated cup-like structure called Bowman's capsule. This glomerulus is formed by the capillaries that arise from efferent arterioles, and these glomerular capillaries drain into the efferent arterioles. So we can see that these glomerular capillaries are interposed between these afferent arterioles and efferent arterioles. And these capillaries are the only capillaries that start with arterioles and end in arterioles. That is, the vascular system in the glomerulus is purely arterial. Another thing to be noted here is that the diameter of efferent arteriole is less than that of afferent arteriole. When we look into the histology, we can see that the glomerular capillaries are formed of single layer of endothelial cells which are attached to the glomerular basement membrane. And this glomerular endothelium are fenestrated, that is, they are having pores in between, as you can see in the picture. Now, let's study the structure of Bowman's capsule. Bowman's capsule is the initial cup-like dilated portion of the nephron that accommodates the glomerulus. It has got two layers, the inner visceral layer, which is continuous with the outer parietal layer at the visceral pole. So this visceral layer is in close contact with the glomerular capillaries enveloping it, and the parietal layer forms the outer limit of the renal corpuscle or the outer portion of the corpuscle, which gets continued as the walls of the tubular portion of the nephron. And there's a cleft-like space in between these two layers called the Bowman space, and this Bowman space is continued as the lumen of this tubular portion of nephron. When we look into the functional histology of Bowman's capsule, we can see that both the layers are having a single layer of flattened epithelial cells which are resting on the basement membrane. The parietal layer of Bowman's capsule is formed by parietal epithelial cells, and visceral layer is formed of the cells called podocytes that are having feet-like cytoplasmic extensions called pedicles. And these pedicles are arranged in an interdigitating manner, having a small space in between. We have already learned that the glomerular capillaries are formed of a single layer of endothelial cells that are arranged on a basement membrane, and these glomerular capillaries lie much closer to the inner visceral layer of Bowman's capsule. So what happens is that the basement membrane of these glomerular capillaries fuses with the basement membrane of visceral layer, and thus this fused basement membrane forms the separation between this glomerular capillary endothelium and the visceral epithelial layer. The capillary endothelial cells, basement membrane, and podocytes of the visceral epithelial layer together forms the filtration barrier. We have seen that the capillary endothelium is fenestrated. They are also negatively charged because of the presence of anionic glycoproteins. Therefore, the negatively charged protein molecules are not easily filtered through this filtration barrier. Hence, protein is absent in urine in normal conditions. Likewise, basement membrane, which is a meshwork of fine fibrils in a gel-like matrix, also prevents the filtration of proteins. Then podocytes, which form the inner layer of the filtration barrier, are having cytoplasmic extensions called pedicles, which are arranged in an interdigitating manner, having slit pores in between. They too do not allow free passage of molecules. Thus, filtration of molecules is greatly restricted by this filtration barrier, especially in case of protein molecules. We can see that the filtration through this glomerular-capsular filtration barrier occurs through the extracellular route, that is through the pores in the capillary endothelial cell layer, basement membrane, and slit pores in the podocytes. The molecules with effective molecular radius of 20 to 45 angstrom and cationic molecules are easily filtered through this barrier. But in certain conditions, the permeability of the glomerular capillaries may be increased. That will lead to proteinuria, that is the condition in which proteins are present in urine, as in case of nephrotic syndrome. Next is mesangium, which is an important component of renal corpuscle. It consists of mesangial cells and mesangial matrix. The mesangial cells which are present in the glomerulus closer to the glomerular capillaries are called as intraglomerular mesangial cells. The important functions of mesangial cells are: they provide structural support to the glomerular capillaries, they secrete extracellular matrix, they secrete the prostaglandins and cytokines, and they are phagocytic in nature. The mesangial cells contain contractile units which are present in close contact with the glomerular capillaries, helping in alteration of the glomerular filtration rate. The mesangial cells that are present outside the glomerulus are called extraglomerular mesangial cells or Lassus cells, and they secrete some quantity of renin and erythropoietin. Now comes the tubular portion of nephron, which is comprised of proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Now, let's see the peculiarities of proximal convoluted tubule. As it is the initial segment of the nephron, it is called as proximal tubule, and since majority of it is coiled, it is called by the term proximal convoluted tubule. And it ends in a small straight segment which continues to form loop of Henle. So the initial major convoluted portion is called by the term pars convoluta, and the distal smaller straight portion is called as pars recta. The proximal convoluted tubule is about 15 millimeter long and 40 micrometer diameter, and is formed of brush-bordered cuboidal epithelial cells. This proximal tubule recovers most of the glomerular filtrate for its considerable capacity of reabsorption. So, the pars recta of proximal tubules continues with the loop of Henle. Loop of Henle consists of descending and ascending limbs. As we have already discussed, irrespective of the position of nephrons, that is whether cortical or juxtamedullary nephrons, the descending limb is thin throughout all the nephrons, whereas the ascending limb has thin and thick portions in a typical juxtamedullary nephron, but a cortical nephron has only a thick ascending limb. So the cortical nephron will be having a thin descending limb and a thick ascending limb for the loop of Henle, whereas juxtamedullary nephron will be having a thin descending limb, a thin ascending limb, and a thick ascending limb. The total length of loop of Henle varies from 14 to 26 millimeter, and about 15% of the filtrate is reabsorbed in this loop of Henle. So the loop of Henle of juxtamedullary nephrons play an important role in concentrating the urine. When we look into the peculiarities of thin limb of loop of Henle, we can see that in cortical nephrons, the thin segment is very short, has no ascending portion, and it barely penetrates the medulla. Whereas in juxtamedullary nephrons, the thin segment is long, has an ascending portion, and penetrates deeper into the medulla. The diameter of thin limb of loop of Henle is about 15 micrometer. It is formed of flat epithelial cells without brush border, which suggests the passive exchange of solutes and water in this part. So from the thin limb of loop of Henle to the thick limb of loop of Henle, we can see there is an abrupt transition between these segments. The diameter, the tubular diameter increases from 15 micrometer to 30 micrometer, and it is made of small cuboidal epithelial cells without any brush border that suits for active transport of solutes and water. The thick ascending limb of loop of Henle terminates in the distal convoluted tubule, and the junction between the thick ascending limb and distal convoluted tubule comes in contact with the afferent and efferent arteriole. There lies this specialized structure called the juxtaglomerular apparatus. And here the tubular epithelial cells are modified to form macula densa. So the distal convoluted tubule begins immediately after the macula densa. It is about 5 millimeter in length, and this DCT is formed of epithelial cells which closely resemble the cells in the thick ascending limb, that is cuboidal epithelial cells without any brush border. But the cells in DCT are comparatively larger in size. Also, a few intercalated cells with microvilli are present here. This segment is relatively impermeable to water, but hormones like aldosterone and antidiuretic hormone can facilitate the reabsorption of water from this segment. Collecting duct. The distal convoluted tubule empties into the collecting duct through a small and relatively straight tubule called connecting segment. Several such DCTs empty into the collecting duct, which passes through the cortex and medulla and empty into the renal pelvis at the apex of medullary pyramids. Thus, the collecting duct has got two parts, cortical portion and medullary portion. The cortical portion is called as the arched collecting duct, and the medullary portion is called as the straight collecting duct. The medullary portion is further divided into outer medullary part and inner medullary part. The collecting duct is formed of cuboidal or columnar epithelium, which are modified to participate in the transport of ions and water. So the two types of modified epithelial cells in the collecting duct are principal cells or P cells and intercalated cells or I cells. The P cells are involved in sodium reabsorption and ADH-induced water reabsorption. The I cells contain more microvilli and are involved in acid secretion and bicarbonate transport. Here in this slide, the histological specialties of different parts of nephron that we have already studied in the previous slides are being summarized in a single table. This shows that the Bowman's capsule is formed of flat epithelial cells, the proximal convoluted tubule is formed of brush-bordered cuboidal epithelial cells, the thin limb of loop of Henle is formed of flattened epithelial cells without brush border, the thick limb is formed of cuboidal epithelial cells without brush border, the distal convoluted tubule is formed of cuboidal epithelial cells without brush border, and the collecting duct is formed of cuboidal or columnar epithelial cells that are modified into the principal cells and intercalated cells. Here, the passage of urine is summarized. The straight collecting ducts from the medullary pyramid unite to form papillary ducts or ducts of Bellini, and they open into the V-shaped area called papilla. So the urine which was collected in the collecting duct will be drained into this papilla, and from there it will be drained into the minor calyx. 3 or 4 minor calyces unite to form a major calyx, and thus from the minor calyces urine passes through the major calyces and reaches the pelvis of the ureter. Pelvis is the expanded portion of the ureter present in the renal sinus, and from the pelvis through the remaining portion of the ureter, the urine passes downwards and reaches the urinary bladder. Thank you for your patient listening. I hope you all are safe and secure at your houses with your parents and dear ones. So along with these studies, try to enjoy these precious moments that you got by chance. Stay home, stay safe.